A very good morning, friends. In this video we are going to study about an important topic of carbohydrate metabolism, that is glycolysis, under the headings of important aspects, steps, regulation, energetics. Glycolysis is also known as Embden-Meyerhof-Bernas pathway. Now coming to the steps. Glucose is converted to glucose 6-phosphate. Glucose 6-phosphate is converted to fructose 6-phosphate. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Now this is cleaved into 2,3 carbon compounds, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is converted to bisphosphoglycerate. Bisphosphoglycerate is converted to 3-phosphoglycerate. Three-phosphoglycerate is converted to 2-phosphoglycerate. 2-phosphoglycerate is converted to phosphoenol pyruvate, and phosphoenol pyruvate is converted to pyruvate. Now the steps in detail. Glucose is converted to glucose 6-phosphate by phosphorylation reaction by the action of enzyme hexokinase or glucokinase. 1 ATP is utilized in this reaction. Glucose 6-phosphate is converted to fructose 6-phosphate by isomerization reaction by the action of enzyme phosphohexose isomerase. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate by phosphofructokinase. Now this fructose 1,6-bisphosphate is cleaved by aldolase into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now phosphotriose isomerase catalyzes the reversible interconversion of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now glyceraldehyde 3-phosphate dehydrogenase brings about the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. Now bisphosphoglycerate kinase brings about the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate. 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase, and 2-phosphoglycerate is converted to phosphoenol pyruvate with the help of enzyme enolase. Phosphoenol pyruvate is converted to pyruvate by the action of enzyme pyruvate kinase. Now, in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, 1 ATP is utilized. Now from this glyceraldehyde 3-phosphate, there is generation of NADH. From NAD, with the help of glyceraldehyde 3-phosphate dehydrogenase, this NADH in electron transport chain and oxidative phosphorylation gives 2.5 ATP. Since there is production of 2 molecules of NADH from 1 molecule of glucose, it gives 5 ATP. Now in the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, this is a substrate level phosphorylation reaction. There is production of ATP from ADP without the involvement of electron transport chain and oxidative phosphorylation. Now phosphoenol pyruvate is converted to pyruvate with the help of enzyme pyruvate kinase. This is also a substrate level phosphorylation reaction. There is production of ATP without the involvement of electron transport chain and oxidative phosphorylation reaction. Now coming to the regulation. Basically, 3 enzymes are involved in regulation: hexokinase or glucokinase, phosphofructokinase or pyruvate kinase. Hexokinase is inhibited by glucose 6-phosphate. Glucokinase, it is activated by insulin and inhibited by glucagon. Phosphofructokinase is induced by insulin and repressed by glucagon. Since it is an allosterically inducible enzyme, it is allosterically activated by AMP and fructose 2,6-bisphosphate and allosterically inhibited by ATP and citrate. Pyruvate kinase is activated by insulin and inhibited by glucagon. Now coming to the energetics. In this aerobic glycolysis, glucose is converted to pyruvate, and in aerobic glycolysis there is production of 7 ATP. From glucose molecule we have glyceraldehyde 3-phosphate, and from glyceraldehyde 3-phosphate we have 2 molecules of NADH, and from NADH it gives 5 ATP. Now from substrate level phosphorylation reaction we have 4 ATPs. From one molecule of glucose we have 2 molecules of NADH, and from substrate level phosphorylation reaction we have 4 ATPs. Now there is consumption of 2 ATPs, so net we get 7 ATPs. Now coming to anaerobic glycolysis, this pyruvate is converted to lactate with the help of enzyme lactate dehydrogenase. Now from glyceraldehyde 3-phosphate step we use NADH molecule, and this NADH molecule is utilized to produce NAD, and this NAD is utilized in here. So in anaerobic glycolysis we have 2 ATPs production, 4 ATPs from substrate level phosphorylation reaction and 2 ATP consumption, so we get 2 ATPs. So this is all about glycolysis. Please like, share and subscribe. Thanks.